Hello, everybody. Welcome to episode 1 of the AP Biology podcast. I'm Rishi Malik, and in this episode, we're going to be discussing the biological motor that powers life. Thank you guys for tuning in to episode 1 of the AP Biology podcast. Since it's the first episode, I wanted to start out with a banger, so I decided to make this episode about one of the most complex things in the entire universe, to be honest, which is ATP synthase. In this episode, I aim to kind of connect by talking about ATP synthase. I'm aiming to kind of connect this episode to a bunch of units that we had in AP Biology. Throughout this video, I'm gonna have an animation on the screen that's just gonna be kind of like illustrating everything that I'm saying, because it would be kind of hard to follow what I'm saying without a visual representation. So hopefully that's helpful, and hopefully it makes it less boring. Let's get into this episode. Alright, so we all know what ATP synthase is, right? It's a very important enzyme that produces ATP, which is the universal energy currency for cells, and it does that by harnessing the energy from the proton gradient that is formed in either photosynthesis or cellular respiration. Honestly, you can think of ATP synthase as a tiny wind turbine inside your cells that is generating the energy that your cells use for a lot of the chemical reactions. So just some background knowledge before I start discussing the structure and function of ATP synthase. I want to show you guys the two main parts that make up ATP synthase. The F1 unit on the top is where ATP is synthesized from ADP and inorganic phosphate, and the F0 part has the ring that is rotating on the screen right now, and that ring rotates when protons flow through the membrane, and I'm going to get into that in a sec. In this class throughout the school year, there's this recurring theme of structure and function, and this enzyme is a perfect example of how important the structure of something is to its function in biology. And I went ahead and researched the units that make up the enzyme, which I stated earlier, the F0 and F1 parts, and I wanted to see how they enabled ATP synthase to actually function and produce ATP. And it's actually super interesting, and just talking about that alone connects to the multiple units that we had in this class. So like I said earlier, the F0 part of ATP synthase has this ring, and it's called the C ring. This ring has proton binding sites on each of its subunits, and protons will bind to their proton binding sites, and the C ring will use the energy from the protons, and it will turn it into mechanical energy that it can use to rotate. And that's why I said that this ring is analogous to a wind turbine, right? Because wind turbines do the same thing. They use wind to make mechanical energy to rotate. I'm pretty sure. The protons have a lot of potential energy because they want to do anything that they can to get to the other side of the membrane where there is less protons, and the only way that they can do that is to go through ATP synthase. And as this ring is rotating, it also causes this shaft right above the ring to also rotate. It's called a gamma subunit. It's a shaft that leads directly into the F1 unit where ATP is synthesized from ADP and inorganic phosphate. When this gamma subunit rotates, induces the conformational changes in the F1 unit of ATP synthase needed to catalyze the synthesis of ATP. So yeah, there's a little bit of unit 1 in there. So I think that's really cool how a lot of what we learned in this class can just be directly used to learn about this enzyme. There have been many experiments and studies done about ATP synthase, and I found a really cool one from 2023, and it was done at the Tokyo Institute of Technology, and they actually found out that chloroplasts use a redox regulation mechanism to control the activity of ATP synthase. So remember, redox is when electrons are added and taken away in a reaction, and this mechanism allows ATP synthase to adjust its ATP production in response to varying light conditions. And remember when we learned about the cysteine amino acid in the beginning of the year? The way that this works is by modifying the redox state of cysteine residues. So remember, something that makes cysteine stand out is the fact that it has a sulfhydryl group, and this functional group can go through reversible oxidation to form various different acids, which alters the protein's structure and function. And this redox sensitivity allows cysteine to act as redox sensors and also act as switches in the signaling pathways in the cell. And what that means is when the cell's environment changes during stress or signaling events, the cysteine can either get oxidized or reduced, which would trigger conformational changes in proteins. And these conformational changes could then turn certain processes on or off like a switch. I felt like a good way to end this episode was to talk about the evolution of this complex enzyme. Evolution is one of my favorite topics ever, so I was excited to do some research on this. The evolution of ATP synthase is super interesting. So ATP synthase is classified into two main types. The divergence of these types potentially predated the split of archaea and bacteria, which means that the divergence of ATP synthase may have occurred over 4 billion years ago. It's also thought that the head of ATP synthase, so like the F1 part that I talked about earlier, evolved first, again over 4 billion years ago. And obviously back then, over 4 billion years ago, ATP synthase obviously wasn't as advanced as it is today, and that's pretty easy to tell because we know that for billions of years organisms were anaerobic, produced very little ATP. I don't know for sure, but I think that maybe when the oxygen levels started to rise on Earth, that made ATP synthase more useful, and it was actually able to function better in the presence of oxygen. So since organisms were now getting more ATP, life was able to be more complex. Something that played a pretty important role in the evolution of ATP synthase was endosymbiosis. I actually found out that the evolution of ATP synthase in chloroplasts can be traced back to an endosymbiotic event where a cyanobacteria was engulfed by a bigger cell, and this endosymbiosis event ultimately led to the first photosynthetic eukaryotes and the chloroplasts within them. The endosymbiosis of ATP synthase was super important for the evolution of it because it allowed ATP synthase to spread across all domains of life, which is archaea, bacteria, and eukarya. Before this episode of the podcast ends, I just wanted to discuss some real-life applications of ATP synthase. So obviously ATP synthase is important for our health. It helps us maintain homeostasis and overall health. However, defects or mutations in ATP synthase can lead to various diseases. I'll discuss a few. So a defect in the ATP6 gene in mitochondria are linked to neuropathy and maternally inherited Lee syndrome. There's obviously a lot more diseases and conditions that are linked to ATP synthase mutations, but that could be a topic for a whole other episode for the podcast. There are a lot of neurological disorders that are linked to ATP synthase defects though. Secondly and lastly, ATP synthase can actually be used in bioengineering and biotechnology. So the rotational mechanism of ATP synthase could potentially be coupled to signal transduction, which would allow scientists to use it in developing bioelectronic devices like biosensors and biochips. And ATP synthase from thermophilic organisms would be used for experiments and research on these biotech ideas, because thermophiles live in very high temperatures, so the stability of their ATP synthase would allow it to function in a variety of environments that researchers and scientists can use. Thank you all for listening to episode 1 of the AP Biology podcast. Overall, I would rate this enzyme a solid 9.8 out of 10. I find it really interesting how complex it is, and I also find it crazy how an enzyme evolved into something this complex, and now I can also finally understand why this is Mr. Garn's favorite enzyme. Thank you all. I hope to see you in episode 2 where we discuss nitrogen fixation. Thank you all for watching. We hope to see you in episode 2 where we discuss nitrogen fixation in bacteria. I hope you all have a great summer break. Thank you.